Okay, Mark, can you see me now?
Yeah.
It looks like all the committee members are here. So,
okay, great. Hi, everybody. We're going to get going. Welcome to Mike Brawner's PhD defense. This is a big day, and I'm excited that we have reached this day and that I get to introduce Mike to you now. So Mike arrived at OSU in the fall of 2012, which was actually the exact same term that I arrived at OSU. He was an undergrad transferring in from Portland Community College, and then he earned his bachelor's at OSU in zoology in 2014. So I've known Mike for a really long time, basically since I first arrived at OSU. So you may not know this, but Mike actually had a previous career as an accomplished chef before returning to school to pursue his interests in biology, ecology, birds, natural history, you name it. So I didn't actually meet Mike until the fall of 2013. And I met him because he enrolled in my By 370 Ecology large lecture class. And it can be hard to develop a close relationship with students in that class because it's large. It's over 200 students, but he totally knocked my socks off in that class. As a side note, it's probably good that you waited till 2013 and didn't do that in 2012. Since it was less than smooth its first time through. Otherwise, we might not have ended up here today in this moment. So Mike was really intrigued by the work we were doing in the lab. So he came on board shortly after taking ecology as a lab assistant. He was also working for Paul Bradley at the time in the Blaustein lab. So he came highly recommended from the 5th floor. And then he enrolled in my paleo class in the spring of 2014, which is a much smaller class where we spend a lot of time together in the field. So that's where I really got to know Mike and his interests. He was very interested in pursuing a graduate degree, and he was already an integral part of the lab. So he was operating more like a grad student than an undergrad, basically from when I first took him on as an undergrad in the lab. So it was a total no-brainer to have him join us as a grad student. I was thrilled that he wanted to stick around and officially join us in 2015. He spent a little bit of time down in Portal, Arizona that summer working as a field assistant, and then came back and began his graduate studies in the IB department. So, like I said, Mike has been with us in the Terry Lab essentially from the beginning. He's a fixture. So this really feels like, it's marking the end of an era to have him up here presenting his work to you today. I've known him for a long time now, and he is always calm, always thoughtful, ever supportive. He's just a fabulous colleague, a fabulous mentor to students in the lab, and he's also been a really wonderful friend to have around through all of these years. So as Mike will tell you, it's been a push here at the end to get across the finish line. This last year and a half of pandemic life marked by isolation and stress from the pandemic on top of the normal challenges of finishing. That's been hard for lots of people. And I am incredibly proud that Mike is here today doing this. He's done it. He's worked extremely hard to get to this point in time through this really adverse set of challenges. And I'm just so impressed and proud of all that he's accomplished with his work. So as for his research, well, he's going to tell you about it, but I just want to say a couple of things. Mike, to me, strikes me as just sort of this endlessly creative, deep thinker. He reads widely. He integrates lots of ideas across traditional research boundaries and always has interesting things to talk about from things that he has learned. The research that he's going to present to you today, it is truly and fully his own. He developed these questions and the approaches, all of this was honed and implemented by him. So, he's in his time in our lab, he has turned into our resident expert in macroecology, biogeography, global change biology. And so let's see, what else about Mike? He's also deeply committed to making each space he inhabits a better place for everyone in it, a more equitable place, more diverse spaces. And we all have benefited from that work as well, both at the department level, from his time in student government or governance and his work on the EJI committee, as well as a lab in terms of his interactions with all of us in the lab. So I really want to explicitly commend him on the work that he has done in that arena as well. And let's see, and his baking skills. I would be remiss if I didn't commend him on his culinary skills. My goodness, the lab was a better place because of the supply of home baked delicacies that he would drop off in the lab, sometimes for lab meeting, but sometimes just because he had baked. So in sum, I guess, I never cease to learn things from Mike. It's my utmost pleasure to introduce him to you today as he defends this impressive body of work that is his dissertation. And before I turn it over to him, I was told by Teresa to say that questions, so we'll open it up for questions at the end. And given the remote format here on Zoom, the best ways to ask a question will be to raise your hand or put a question in the chat. And both Teresa and I will monitor those and make sure that the question reaches Mike. So without further ado, Mike, it's all yours. Good luck.
All right. Well, thanks, Becca. Thanks for that wonderful introduction. Wow, that's a lot to live up to. But here we go. So thank you all for joining us today. So I'm going to be sharing what I've been doing over my dissertation for the past 6 years. And again, my name is Michael Brawner, and I'm going to talk to you about how context and scale mediate the impact of climate on a widespread avian predator, the American kestrel, which you see here on your screen. Now, before I begin, I want to give you a quick outline for today's talk. So I'll start by giving you some background information and some of the scientific motivations for my work. Then we'll look at how climate and other environmental variables impact nest success in American kestrels. We'll pull out a little bit and start to look at the species scale and think about how we measure body size in birds and what do we mean when we talk about body size. And then lastly, we'll look at geographic and typical variation size in the American kestrel. So the Intergovernmental Panel on Climate Change just released its sixth report this year. They show that human-caused climate change has increased the global surface temperature by an average of 1 degree Celsius in 1850. the last 4 decades have been successively hotter than any other. We know that we continue to increase temperature 2 degrees warming under the best case scenarios of mitigation, and potentially even higher. Okay, now the impacts of this warming are widespread across the globe and include things like increasing drought, increasing frequency and magnitude of severe weather events such as hurricanes, an increased risk in wildfires, ocean acidification, and sea level rise. And a lot of these problems are things that we are all trying to deal with currently. The documented impact on wild species has also been great. We see impacts play out across a wide diversity of species and ecosystems, both in marine and freshwater ecosystems, and in a variety of terrestrial ecosystems as well. Now, the possible responses of animals to this rapidly changing environment can be lumped into 3 main categories. Animals can move from the impacted region, they can adapt in place, or they can go extinct. 2 of the most common responses among animals, shifts in species ranges towards higher latitudes and altitudes, and advances in phenology, are so well documented now to be thought of as universal responses. Now, phenology is just a term ecologists use to talk about the timing of life history events. So think about trees leafing out in the spring, or the arrival of birds on their breeding grounds. In fact, we can use birds to look at the support for these responses because birds are so well studied and because the response is so coherent across a wide range of taxa and across many different regions around the globe in birds. So 2 studies of birds in the northeastern United States really highlight these responses. The first here is from Rushing et al. In 2020, and they showed a significant expanse in the northern range margin and in the mean breeding latitude of 32 different breeding birds since about 1986. And this corresponds closely with sharply rising temperatures in that region. An earlier study found a close link between mean temperature and the arrival of birds to their breeding territories. So birds arrived earlier to the breeding grounds when the temperature was higher, showing the advance in nesting phenology here. Now, both of these patterns are occurring in birds in northeastern North America, but these patterns are seen throughout a wide variety of species and across the globe. A third universal response has also been recently proposed. These authors argued that increasing global terrestrial and sea surface temperatures could select for smaller individuals because they are better able to thermoregulate in a hotter climate. However, as we will see and discuss later, the support for this is equivocal currently. Now, I want to take a step back and look at the broader context of bird populations, because it's not just climate change that is impacting bird species. So as you can see in this graph here on the left, it's been estimated that approximately 3 billion birds have been lost since 1970 in North America alone. Now, the graphs on the right show that this loss has not been equivalent across different biomes, and that grassland birds have been particularly impacted. Industrial urbanization, our increasing reliance on toxic pesticides, and industrial agriculture have certainly been implicated in this loss, but the impacts of climate change no doubt compound the effects of these other threats to bird species. It may surprise you to learn that some of the species experiencing the greatest losses are also some of the most abundant species in North America. Out of the top 10 species with the greatest losses, many of them are species that most of us would recognize as abundant and common. 2 of these, the European starling and the house sparrow, are actually invasive species. Now, setting aside the negative impact of invasive species, the loss of ecosystem services that occurs in tandem with losing such magnitude of common birds is a real concern. Common species have their greatest effect through their numerical abundance, and when that diminishes, the benefits to ecosystems diminishes. Now, one historically abundant grassland species that has been in decline over this time period is the American kestrel. It's the smallest falcon in North America, and kestrels are diet generalists that primarily hunt in open habitat such as grasslands or other sparsely forested areas. It is a widespread species occurring across the Western Hemisphere. So in this map, the range of kestrels is in this light yellow color. And there are an estimated 17 subspecies that occur throughout this range. Now, it's also a sexually dimorphic species, meaning that females like this bird on the left here and males like the one on the right differ both in size and plumage coloration. And maybe it's partly because of their habitat of hanging out on power lines and hunting by roadsides, the kestrels are seen as quite common and ubiquitous. However, we know from breeding bird survey data that kestrels have been experiencing a long, slow, steady decline since the late 1960s. They've seen about a 54% population decline over this period. Now, the usual suspects such as habitat destruction, climate change, and rodenticides have all been suggested as possible causes, but to date, there's just not any clear evidence for one of them, and it's likely a combination of multiple causes that we haven't quite got a grasp on yet. Now, to put this decline in perspective, we can look at the loss of other diurnal raptor species over the same period. Looking at just this group, American kestrels make up 82% of the loss of this group by number of individuals. When we look at biomass, kestrels represent nearly half of all biomass of diurnal raptors lost since the 1970s. This is a lot of kestrels no longer feeding on the landscape. There is also some evidence that kestrels are impacted by climate change in similar ways that we have seen from other species. So for instance, at a long-term breeding site in southwestern Idaho, the nesting phenology there has increased by an average of 15 days over a 23-year period. Evidence here suggests that kestrels are responding to warmer winter temperatures and an earlier start to the growing season, particularly in agricultural habitat. There's also some suggestions of declining body size in kestrels. So data analyzed from a network of migration banding sites found a significant decline in both mass and wing chord at several of the sites. All right, so that brings us up to my first analysis chapter, chapter 2 of my dissertation. When I first started grad school, Becca introduced me to a family friend of hers who has been monitoring kestrels and nest boxes over in Central Oregon for many years. And I started working with them and became interested in asking questions about how climate and other environmental factors are impacting the nest success of kestrels in that population. So I also compared that data to other populations once we got results from that analysis. And we also looked at the spatial scale of analysis to see if that had any impact on our results. So the environment can play a crucial role in the nest success of birds. We've already discussed how climate can impact the timing of breeding, but extremes in temperature and sustained heat can also affect adult and nestling birds through either direct mortality or by lowering the condition and foraging efficiency of adults. Studies of nestling kestrels in Alberta, Canada showed they had a higher probability of mortality when there was more inclement weather. So that's shown on the x-axis of this graph here, more inclement weather on the right side and the probability of mortality on the y-axis. The composition and structure of the landscape too can influence nest success by altering the microclimate of the nest, for example, or by determining which predators have access to the nest through processes of fragmentation or other landscape processes. The recent work on small mammals show that bottom-up influences on small mammal population dynamics through climate-induced variation in primary productivity may be stronger than previously thought. Now, small mammals are a critical diet resource for American kestrels. Other work on primary productivity looking at ducks showed that ducks had a lag response of about a year after a primary productivity pulse. So they didn't see increased nest success until a year after there was a pulse in primary productivity. So there could be some lag effect as that those bottom-up influences filter up through the food chain. Considering all these factors, we set out to determine which of these might have an impact on nest success in kestrels breeding in Central Oregon. We were lucky enough to team up with some of the community scientists that I mentioned earlier and the Sisters area that had been monitoring kestrels and boxes over there for quite some time. Now we helped them ban nestlings and collect data for a few years, and from that work and previous efforts, we were able to build a 7-year nest history for this population. We ended up with 493 observations. For each of these, we recorded the fate of the nest and the days since the last observation. And with these key pieces of data, we were able to build statistical models and compute a daily survival rate for each nest, which we could then translate into a nest success rate, or the probability that at least one nestling will fledge from that nest. So for the environmental variables, all of these were derived from satellite imagery. So we used a landfire data set to gather our landscape variables. We looked at both composition and structure. Now, to understand the difference between these two, you can think about composition as the color in the two maps I'm showing here. They both have green, but they have different amounts of green in them. The landscape structure then is the pattern in which those colors are put together. So is the green color more aggregated or is it more dispersed? We also collected data on primary productivity. So we used the NASA MODIS Terra data for this. And we also collected climate data as well through the PRISM group here at Oregon State University. So average, mean, max temperature and cumulative precipitation for both annual and seasonal measures. So for all of these environmental variables, for each active nest box, we then calculated an average measure at various spatial scales, so at the 1, 5, and 10 kilometer spatial scale. And then we replicated this procedure for the 2 previous years to determine if there was any lag effect on nest success. We also calculated some seasonal variables. So initiation date, which is the when the first egg was laid in the nest, relative to the start of the breeding season. And here we classified the start of the breeding season as May 1st in our population. That was sort of the average date throughout the time period we were studying. We looked at nest age. So this is the age of the nest since the initiation of the nest at the time it was observed. And the observation date too. So the day of the year relative to the start of the breeding season. So for each spatial scale, we took our 36 environmental variables and transformed them into 5 principal components using a principal components analysis. This ordination method allows you to take highly dimensional and highly collinear data sets and reduce them to a few interpretable variables. So we went from 36 variables down to 5 variables, which explained a cumulative 70% of our variation. We then built models based on the logistic exposure method of Schaefer using generalized linear mixed effects models. This framework allows us to account for the number of days a nest is exposed to potential failure and therefore avoid some biases that can happen from finding nests late in the season. And then next, we fit the fate of the nest, either success or failure, to our various combinations of seasonal variables and our 5 environmental principal components. So we also included nest ID and year here as fixed effects in these models to account for the non-independence of repeat observations at the same nest within the same year. And we used the model selection criteria from Burnham and Anderson using Keikki's information criterion corrected for a small sample size. So let's look at some results. So nest success in Central Oregon kestrels was pretty stable throughout the period that we studied. There was no significant difference between the years, although 2013 was a little bit lower. This graph here is showing the probability of nest success on the y-axis and, of course, year on the x-axis here. So, and this was also relatively high when we compared this with populations around North America. Oregon here highlighted at the top at an average of about 75% nest success. So that's good to know. Kestrels in Oregon seem to be doing quite well. The top 2 models based on our model selection results showed that a model with all 3 seasonal variables, or one with the seasonal variables and the first principal component, environmental variable, were the best supported. So this was true for all 3 spatial scales, so we found no influence of spatial scale on the analysis. We interpreted the first principal component as a gradient of greater to lesser primary productivity. Now, one surprising result that we found was that nest initiation date showed an opposing pattern to what is generally established for most bird species. So the graph here is showing nest initiation date on the x-axis, and 0 represents May 1st, the start of the breeding season, and then probability of nest success on the y-axis. And we see that the probability goes up as the nest initiation dates get later in the year. And this is exactly the opposite of what you see for most bird species and even other kestrel populations. So there seemed to be some sort of cost associated with early nesting in our population. Now, the next graph I'm showing here shows PC1 on the x-axis, and again, probability of nest success on the y-axis. And here, higher values or higher primary productivity are represented by lower values of this principal component. So as primary productivity increases on the landscape, so too does the probability of nest success. When we looked at nest initiation date, we found no trend either decreasing or increasing through time, despite the fact that temperature was increasing in our study. So no advance in phenology in our population. However, when we did an analysis of variance, we found that there were significant differences between some years. So for instance, 2015 had earlier nest initiations than either 2012 or all 3 years between 2018 and 2020. And 2016 similarly had earlier nest initiation dates than 2012. Okay, to sum up this project, nest success in Central Oregon kestrels is relatively high compared with other populations. However, it's important to note that nest success alone does not equal population growth or stability. A recent study actually showed that adult survival and immigration are likely more important vital rates for kestrel population growth. We didn't find any advance in nesting phenology, despite the similarities in habitat and latitude with other populations nearby showing advances in phenology. The one hypothesis for this is that late winter storms and continued snow cover into the spring could prevent kestrels from advancing in nest initiation. So another recent study found snow cover to be a significant factor in delaying spring arrival of kestrels across a wide range of latitude in North America. This might also help explain the cost associated with the early nesting that we saw in our population. We also saw that seasonal measures might be more important than some of those broader scale annual averages and climate that we were using. So seasonal measures likely reflect more reflective of daily weather patterns or extreme weather events or other biotic interactions like predation, which we know has a huge influence on nest success in birds. However, we were not able to catalog that in our study. Okay, now we're going to move from the scale of a single population to thinking about American kestrels across a much broader range of the species. So during my dissertation, I was really interested in addressing questions pertaining to biogeography and macroecology. And the question of whether animal size is responding to climate change seemed like a really interesting topic to investigate. So given that American kestrels have such an expansive range, it is a species well suited for tackling these topics. But before diving into answering questions about geographic and temporal size variation, I first had to address the more fundamental questions related to how we measure size in birds. What do we mean by size when we talk about ecogeographic rules? Again, how do we measure it? And what data best represent structural size in museum specimens? So it's been said of body size that it is the single most important axis of biodiversity. Because of its consistent scaling with metabolism, body size sets the pace of life in all organisms. It is this fundamental feature of body size that makes it such a useful trait in ecology. Many physiological, ecological, and evolutionary processes can be explained in terms of body size. And this is exactly what the metabolic theory of ecology attempts to do in relating metabolism to body size and temperature. So as a universal response to climate change, shifting species body sizes could have huge impacts on the functioning and stability of ecosystems. Now, ecogeographic rules are something that every intro ecology student learns. I learned it when I took Becca's intro ecology class way back when. So there are many of them, and they all attempt to explain an observed geographic pattern in the variation of a trait. So for instance, Bergmann's rule explains the observed pattern of larger size at higher latitudes or in colder climates. The original hypothesis for this observation was that larger animals have a lower surface area to volume ratio, and so they can retain heat better than smaller animals. It would therefore be advantageous, the reasoning goes, to be larger in a colder climate, hence the observed pattern with latitude. This graphic here is showing the difference between species, but the same would apply to individuals within the species. Seen through the lens of global climate change, Bergmann's rule is hypothesized to look something like this, where body size decreases through time, and the smaller species now find evolutionary advantage in a warming climate. So mass is often used as a measure of body size, as we saw on some of the previous graphs. When highlighting the link between metabolic processes and size, this is likely an appropriate measure. However, in studies of geographic and temporal size variation, mass has the potential to be misleading. To understand this problem, we can think about a bird measured at 2 time points throughout the year. In the first scenario, the bird is measured upon arrival at the breeding ground. It's emaciated from the long migratory flight and completely depleted of its reserves. In the second scenario, a bird is measured just before leaving the breeding ground. Now it's fattened up for the long flight home, and it is completely full of reserves at this point. So mass is a highly labile trait, and it can vary depending on the time of year, as we've just shown, breeding condition, the amount of available resources in the habitat, or for many other reasons. In short, it is a reserve-dependent measure. In order to accurately compare size among individuals across geography or through time, we need to compare the structural size independent of reserves. Now, 2 are commonly used measures for body size and birds weren't mentioning here. Wing chord length which is the length from the wrist to the tip of the longest primary feather, and tarsus length, which is a measure of the tarso-metatarsus bone in a bird's leg. Both of these measures are commonly used as proxies for body size in birds. However, wing chord, much like mass, is reserve dependent. Feathers are molted either annually or twice annually, depending on the bird species. And the length of the wing is also under selection pressures due to flight efficiency that could be decoupled from thermoregulatory mechanisms. Tarsus has been suggested as the best single measure for body size, since it's at least based on a skeletal element and therefore less likely to fluctuate over the course of a bird's life. But those same authors have argued that single measurements should be avoided as indicators of structural size. So in chapter 3 of my dissertation, I argue that the sternum of the bird is a robust measure of structural size. It is the largest, most three-dimensional element of a bird's skeleton. And as a skeletal element, it is largely fixed at adult size and therefore reserved independent. The dimensions of the sternum help define the volume and body cavity of the bird. And it also provides a structure for the largest muscle in a bird's body, the pectoralis muscle, which is a major source of heat generation. So volume and thermogenic capacity are two critical pieces of information in assessing Bergmann's rule, and thus the sternum would appear to be a robust measure of structural size in birds. So it would be great if we could just go out and measure a bunch of sternums to study size variation in kestrels. However, sternums are not frequently cataloged in natural history museums. What we do have are a wealth of bird study scans. Now, millions of study scans are cataloged throughout the world's natural history museums, and these specimens provide a library of historical biodiversity and a lens into the past on the changing physical and genetic characteristics of a species. If we could use study scans to estimate structural size, we could more accurately study size variation in a species. And so that's what we attempted to do, to relate various measurements in the study scans to structural size as measured from the bird's sternum. So we scoured the museum databases and looked for paired study skins and sternums from the same individual. And out of the 52 or 100 or so that we found, only 24 we were able to get our hands on that were actually useful for our analysis. So you can see that the skeletal elements in the sternums are quite rare. So we started by collecting measurements from the study skins. So we've already talked about a few of these measurements, wing chord and tarsus. We also measured tail length, bill depth, and bill width. To characterize a sternum, we used a 3D geometric morphometric approach, starting with 3D imaging of the sternum. We then took these images and used the program SlicerMorph to place homologous landmarks and semi-landmarks on the sternum image. Now, homologous here just refers to a evolutionarily derived landmark or trait. And so all of these red points you can find on any bird, even if they're not in the same exact place. And these semi-landmarks help to define the perimeter of the portion of the sternum here that is in contact with the flight muscle. So from this, we could then calculate a measure of centroid size. So centroid size, to calculate this, you take the distance from all of these points to some central point, you square those distances, sum them together, and then take the square root of that. And that provides a measure of the overall size of the sternum, what we're going to refer to as structural size. For our statistical analyses, we used ordinary least squares regression. And again, so here we had centroid size as our dependent variable, and now all of our study skin measurements, different combinations of our study skin measurements as our independent variables. So we ended up with 124 models that we then compared through model selection, again, using Burnham and Anderson criteria here with AICC. So what did we find? So this is just looking at individual measures of traits. On the y-axis, you see the centroid size of the sternum for all of these. And on the x-axis, you see different traits, single traits as measured against that centroid size. So we only found a relationship, a significant relationship in tail length with centroid size. But you can see if you separate the data by sexes here, that relationship is really just shown in the males. Although the combined relationship here with centroid size is explained about 36% of the variation. So none of the traditional measures of size that we looked at showed any relationship, significant relationship, with what we're determining as structural size. So when you combine several measures of size from study skins, you get a much stronger relationship. So here, the amount of variation explained by the independent variables for centroid size is twice what it was from any single variable. So this is probably our best method then for cataloging structural size from study skins. Some combination of traits that includes tail length, tarsus length, wing chord, and some bill measurements. All right, so in American Kestrels, there's strong indication that traditional measures of body size do not adequately capture structural size, which is what we argue that you should be trying to determine and assessing ecogeographic rules. And we did have a small sample size, and these specimens were limited to a few geographic regions. And we caution that this approach is likely to be species specific. In terms of tail length being the best single measure of structural size, it's a bit unclear because tail length should have all the same caveats as wing length, given that they're both just measures of a feather. So we did consider that tail length could be particularly important for a mode of foraging that kestrels do called hover hunting. Now, this mode of flight is particularly energetically expensive and requires a lot of stability. Animal research suggests that the tail is providing a lot of work and stability at low flying speed. The one hypothesis then is that the tail is optimized to the structural size of the bird in order to assist in this mode of flight. And I have a video here of kestrel hover hunting so you can see what this looks like. So as you're watching this video, what I want you to pay attention to is the tail. And notice how the kestrel fans its tail in and out and moves it up and down and side to side. So you'll see here as the kestrel flies in and stabilizes, its head is barely moving at all. And its wings are providing a lot of the lift. But its tail is really working hard to provide that stability there. So I'll show it just one more time, try to. Let's see if I can go back. There we go. All right, having problems with my PowerPoint here. So, next slide. How do we get to the next slide? Sorry for the delay. Okay, there we made it. All right, so now on to my last analysis chapter. We now have a good method for determining structural size in museum study skins. And so we're going to use that to look at ecogeographic rules in American kestrels. We'll also look at some of the mechanistic explanations behind these rules to see which, if any, environmental or ecological factors help explain the patterns that we see. So we already talked about Bergmann's rule, but I need to define Allen's rule now. So just as a reminder, Bergmann's rule explained the variation in size with latitude and temperature due to thermoregulatory demands. And here's a graph showing moose adhering to Bergmann's rule in Sweden. Allen's rule also deals with thermoregulation. This time, however, the focus is on appendage length, with animals in hotter climates having longer, thinner appendages so that they can shunt blood to these areas and dump heat fast. So a classic example of this is rabbits and hares. And hares that live in really hot climates have these really long, skinny ears and long legs that they can vascularize and dump heat from the body when they need to. And Arctic bunnies and hares have much smaller appendages. You also see this in foxes. They're another prime example of this, where desert foxes have those really huge ears, and then the Arctic foxes have just little nubs for ears to help retain the heat. So just like Bergman's rule, there is speculation that increasing global temperatures could lead to animals with longer appendages over several generations. So the evidence for the climate change effect on size is, like I said earlier, somewhat equivocal. Just looking at a series of titles here from the literature, we see that early on that there seemed to be some support that global climate warming was causing birds to decline in size. Here we can see avian body size correlates with global warming. Global warming and body mass decline in Israeli passerine birds. Declining body size in North American birds is associated with climate change. But then you start to see titles like morphological change in birds not explained by thermal adaptation to climate change. Here, birds seem to be increasing in size due to climate change rather than decreasing in size. And of course, there's a study here showing that birds are kind of all over the place. Some birds are increasing in size and other birds are decreasing in size. So it's really hard to sort of wrap your head around what, if any, is there a general pattern here. Now, a much clearer result comes when we look at bird bills. An emerging area of study has shown that the bird bill is likely serves as a thermoregulatory organ in addition to its job in acquiring food. So these images on the left here show a change in temperature of the bill of a bar-headed goose during a rapid heat load experiment. So the frames were taken 30 seconds apart, and the temperature of the bill increased by about a 10 degrees Celsius change over that period. And here it's likely facilitating the dumping of heat from the body. A study comparing bill size to annual mean minimum temperatures found that 7 out of 8 families of birds adhered to Allen's rule for this trait. And there are many other studies sort of corroborating this function of the bill. Of course, there are other mechanisms of change that can influence a bird's bill or its structural size. So the classic example here being Darwin's finches. In long-term studies of these finches, bill size showed an evolutionary response to the size and hardness of available seeds in the environment. Many authors have argued that food availability, driven by patterns in primary productivity, is the main driver for the clinal increase in size we see with latitude. This graph here is showing the general increase in primary productivity going from white to pink to yellow to green from south to north in the summer months. Geist used this to explain the boomerang type shape he found in cervid body sizes, noting that it correlated closely with the rise and fall of primary productivity over about 60 degrees north. And here he suggested that animals began to get smaller again because of the decrease in primary productivity. Others have argued that biotic influences like competition best predict the change in size with latitude that we see. So when a smaller species in a set of competing species ranges further north than the larger in the set, it can now exploit a greater range of diet items and hence grow larger. So this is in line with the theory of character displacement in ecology. This graph from McNabb shows the increase in mountain lion sizes in this sort of U-shaped polygon with the black circles when they are, once the jaguars are excluded from the range. So jaguars are shown here in the open circles. The theory here is that mountain lions increase as soon as there are no more jaguars in the environment, because they can now exploit a greater range of diet items. So clearly there is a lot to account for here. We started by just measuring a lot of birds. So I visited 9 natural history collections across the Western US, and I measured over 800 study skins. Excuse me. We used the same measurements as before, and we ended up with 562 specimens for analysis, ranging in over 60 degrees latitude from Nicaragua all the way up to Alaska, 112 years, so from 1901 to 2013, and 7 different subspecies, which are labeled here on the map. To calculate overall size, we performed a principal components analysis on our size measures and used a single principal component explaining over 88% of the size variation for the variation in our measurements. We also calculated the surface area of the bill, just simply using the equation for the surface area of a cone, and we corrected this for overall size. Now, again, we gathered environmental data, so both climate and primary productivity data. We used the Climate NA software package introduced by Wang et al. And measured temperature, precipitation, aridity, and seasonality variables. And then we also did the same for primary productivity. We then collapsed these, all of these variables down into just 3 principal components that explain the potential environmental structure that kestrels might experience in their environment. So in order to account for biotic influences, we created 2 competition metrics for our data set. So the first was simply a measure of richness for the possible competitors a kestrel might experience or might encounter at any one location. To do this, we searched the literature for a vertebrate predator bird species that overlapped in range, diet, and habitat with kestrels, and we cataloged these at each specimen location. This map here on the right is showing the number of competitors that kestrels might face based on our criteria across North America. For our second location, our second measure, we created a size distribution of the competitor species at each location. We then calculated the skewness of this distribution, whereby positive values quantified a competitor community, we'll call it, with more similar size to kestrels, and negative values depicted competitor communities that were more distant in size from kestrels. So here we're seeing how the size of the competitors might influence the size of kestrels. So to analyze the data, we used a spatial regression framework with simultaneous autoregressive models. These models add an error term that can account for spatial autocorrelation in the data, which we did find when we tested for it. We then built models with year and latitude and factor terms for sex and subspecies to look for general patterns. And then we added in our environmental variables and competition variables to see if any of those best explain the size patterns that we see. Again, we turn to our old friends Burnham and Anderson here to guide our model selection efforts. So we found a strong nonlinear relationship between size and latitude and adherence with Bergman's rule. But we didn't find any relationship with year. So kestrels, based on our analysis, don't seem to be declining in size or increasing for that matter. But notice the pattern here with latitude. So this is very similar to what we saw with the deer and elk and moose that Geist had drawn a graph of earlier, that sort of boomerang type shape. Now, we didn't find a pattern with year when we looked at the overall data set, but when we broke it down by region, we did see some significant patterns with year. So for size, we saw a significant increase in the Pacific Northwest region, and we saw this increase and then decrease over the last half of the 20th century, early 21st century in the Columbia Plateau region. This map on the right here is showing some of these regions in blue, the Pacific Northwest here, and the Columbia Plateau just to the right of it. Coastal California region down here showed a significant increase in the relative bill area of kestrels. And the Pacific Northwest, again, showed an increase, at least over the second half of the 20th century and early 21st century. Looking now at some of the possible mechanistic explanations for these patterns, we found support both at the continental scale, so this graph on the left, and at the regional scale for our competitor size distribution measure, explaining a significant amount of variation in size. So this lends support here to the theory that size is partly helping to determine, or size is partly determined by biotic interaction. The direction of this relationship here would indicate that kestrels get larger as the average size of competitors they encounter approaches the average size of kestrels. Now looking at bill area, we found a consistent signal of an effect of aridity on the size across several regions. PC3 here indicating a aridity gradient, excuse me, moving from left to right, higher aridity on the right. So at the regional scale, this relationship was nonlinear in several different regions that we looked at. Here, the Mojave region is highlighted in yellow, again, coastal California and Pacific Northwest. But at the continental scale, there was just a simple linear relationship where bill area increased with increasing aridity. All right, so what does this all mean, the kestrel is asking? So in terms of Bergman's rule, we found American kestrels adhere to the rule geographically. So we'll give that a check, but not temporally. So that one gets an X. We found an indication that competitors may play a role in determining size variation in kestrels. Based on the patterns that we saw with latitude and the inclusion of primary productivity variables in the model, we also find a support for the primary productivity hypothesis. So both of these will give a yellow check too. These need more investigation to determine if those patterns hold. In terms of Allen's rule, we do see patterns of increasing bill size over recent decades at the regional scale. And the variation we see is explained in part by aridity. Now, while we don't see any patterns in latitude through time with bill size at the continental scale, we did find that aridity explained the underlying variation at that scale. So Greenberg and Danner found similar results in song sparrows, and others have found patterns of bill size explained by humidity. So there seems to be some support here for a response to climate change in line with Allen's role for kestrel beaks, the kestrel bills. Okay, to wrap it all up, we talked about the potentially compounding effects of climate change. We found that the local context is key in explaining patterns of nest initiation. We suggested a need for a clearer definition of body size and provided what we believe to be an appropriate standard for structural size in birds, and then use that to determine which measures in study skins best represented that measure. And we found support for 2 ecogeographic rules in American kestrels and found coherence across spatial scales for mechanistic explanations for each of these rules. All right, so I have a lot of people to thank for this work. First of all, my committee, Becca, it's been an absolute joy to work and learn alongside alongside such an amazing scientist. You're an amazing writer and teacher, and I've learned so much from you in all of those areas. You've been incredibly gracious with your enthusiasm and encouragement, and I probably would have stepped away from this long ago had you not been there to urge me on. So thank you a million times over. Mark, Rich, and Doug, I can't say enough for the time you've given me over these last few months. Your feedback and direction was just the push that I needed to finish up, and I can't thank you enough. Scott, thanks for stepping in to serve in the role of GCR twice now. I'm so grateful for your time. I've contacted you at the last minute, and both times you were more than willing and eager to help out. So thank you, Scott, for that. To all the members of the Terry Lab, past and present, it has been incredible to work alongside such a dedicated group of individuals. I really appreciate all the support and the feedback on the posters and the many terrible drafts of chapters that I submitted you to. Thanks for all the coffee breaks and lunch breaks and just the great discussions we've had through the years. Of course, our lab couldn't run without the support of many undergraduates and lab assistants we've had through the years. I especially want to thank Riley, Elora, Elia, and Chris for all of their help with my work in the lab. And of course, Bridget, Maura, and Sam too for their support and their help throughout the years. And there's so many other individuals that I'm missing here that have come through the Terry Lab. Thanks to the whole IB community for making this such a welcome and fun environment to work in. A special thank you goes to those of you who do so much extra work on top of getting your PhDs and master's degrees to make this department and university that is welcoming for everyone. I'm continually inspired by the leadership of so many of the grad students in our department. And thank you to the whole IB community for your commitment to the work that we do here and for continuing to provide a healthy space for us to do so. To the 4 Ts in the front office, Tracy, Teresa, Trudy, and Tara, who keep the train rolling for all of us. We could never pull anything like this off without you. Thank you so much for all of your help and everything you do. I want to thank the entire Sisters Kestrel crew, especially Don McCartney, Jim Anderson, and Ken Hoshhagen. They have just been amazingly gracious with their time and welcoming to me and talking me through what they know about the Kestrel population over there. And there's a whole slew of volunteer individuals that spend their time every year going out and monitoring these nest boxes and occasionally maybe getting hit in the head with an angry kestrel from behind. So gotta watch out for those. To all my friends and family out there watching, and those of you maybe tuning in later, I really appreciate all the laughter and everything that you've given me over these last 6 years. Just being able to hang out and get away from the work. I really appreciate having all of you as friends, and I couldn't be happier to have you as such. To the Bernadine Kelly crew, so my partner's family, thank you so much for opening your hearts and your home to me. You guys have been such a wonderful second family to me. I'm really thankful to be a part of your group. To my brothers, Lou and Bo, and their wives, Meredith and Jess, and all my nieces and nephews, thank you so much for your support throughout the years. You guys are great, and I really enjoy seeing you and hope I get to do so again soon. To my parents, who have supported me throughout my life in any endeavor I've been involved in, whether it was going to Florida to visit the Cape Canaveral and the Space Center when I was in junior high, or whether it was flying across the country to visit culinary schools. You guys have always been there for me, and just been a wellspring of support and encouragement. And I can't thank you enough. I really lucked out with the two of you. So thank you both so much. To the two best dogs in the world, Bear and Augie, these guys got me through a lot of hard times over these past 6 years. You know, how much more can I say? They're both really good boys. And to my partner, Gianna, I just can't say enough here. Your curiosity about the world, your kindness, your generosity of spirit, it makes every day spent together so wonderful. I'm so glad we've gotten to share our lives together, and I'm so thankful for all the support you've shown me day in and day out over these last 6 years, and going back even further. I would not even started on this journey way long ago had it not been for you. So thank you. I love you. Yeah. All right, and finally, I couldn't have done this without all the funding I received. So thanks to all the natural history museums that allowed me to come and sample their collections, and to the wonderful funding and grants I've gotten into the integrated biology department for their teaching support. All right, I think that's it. I'll take some questions if you have them.
Great job, Mike. So if you would like to ask a question, you can raise your hand or put something into the chat. And I'm trying to figure out how to see raised hands here. Teresa, if you're there and can help me look.
Yeah. So if you just pop up the participants window.
There we go. Okay.
And then under attendees, yeah. And I'll activate the ability for them to talk. So I'm ready.
All right, the awkward silence of Zoom. Mike, you've got a couple of comments in the Q&A tab. I don't know if you can see those. They're not questions per se, but they are comments. We'll hang on the line here for a few more minutes. So if you have a question, feel free to ask. Otherwise, thanks so much for attending and supporting Mike. And oh, I see a hand from David Taylor.
Hey, Mike, can you hear me?
I can. Hey, David.
Hey, great job. It was really cool to get to see this project like all the way through to the end now. I've seen so many of the pieces as we spent years together in the lab. So great job. I'm curious, when you got into doing the morphometrics on the keel or the sternum of the birds, how did you go about like figuring out which particular aspect of the sternum to really investigate for that morphometric analysis? And did you do a full 3D analysis, or did you focus on one particular aspect?
Mm-hmm. Yeah, thanks for that question, David. So we did do a full, so we did a 3-dimensional analysis on a single side of the sternum, the assumption being that both sides are symmetric. So we just took one side of the sternum to represent our measure of structural size. And we really focused on that area of the sternum that is in contact with the flight muscles. The area that we focused on covers both the length, the width, and the height of the sternum altogether. And so there are some other pieces of the sternum there that are mainly for bone attachment with other bones. And so we eliminated those in our analysis. But yeah, so we did use a full 3D analysis, but it was just on a single side of the sternum. Thanks for that question.
Yeah, can I ask a follow-up to that real quick?
Sure.
So do you know, is there any way to like estimate sort of any metabolic rates or mass-specific metabolic rates from like the surface area that you estimate off of that particular face of the sternum? Or can you get towards metabolic rate in the birds from that? Or is it sort of like taking just that size estimate as a way of speaking about it?
Yeah, that's a really interesting question. So typically, in terms of metabolic rate, mass is often used in conjunction with temperature to estimate metabolic rate. And so this is more a measure of structural size. And so I don't know if you could estimate metabolic rate from that, although it would correspond somewhat to the size of the pectoralis muscle. So there may be a way there to think about that. Yeah, I'd have to spend more time thinking about whether that would be an appropriate measure or whether you just want to use mass.
Okay, yeah, that makes sense. I wasn't certain, but really great talk. It was really fun to see. So thank you for answering that question for me.
Great, thanks so much, David.
Yeah.
You've got another question from Jalen in the Q&A tab. So she says, your talk has me thinking about crossbills, of course. Could you speculate what we could see in terms of Bergman's rule or Allen's rule in the crossbill complex, or really any nomadic migrant?
Yeah, so crossbills are really interesting, especially when you think about the bills, because the function of the bill is so tied to their foraging habit, right? So they have this cross bill that they use to pry open cones. And so it would be a really interesting question, because if it is used as a thermoregulatory organ in crossbills, you would imagine that there would be competing selective pressures there for the thermoregulatory function and the feeding function. So yeah, I don't know right off the top of my head what you might expect to see there, but I imagine in birds where the bill is really a prominent part of the way they feed, that it's going to be less likely to be used as a thermoregulatory organ.
Okay, thanks for that. In the interest of time, given that it's almost 1:15, I think we should end it there. If you have questions about any of this, you can always email Mike and ask him. And it looks like you've got a couple folks who said they're going to catch up with you later anyway for other questions. So I'll just thank everybody for coming again and for cheering on Mike. And Mike and committee, we have a separate Zoom link that I sent you earlier today that we're going to use for the closed door portion of the defense. And so let's take a 10-minute break to get up, stretch, do whatever you need to do. And then we will reconvene in that other Zoom room shortly. So thanks a lot, everyone. And great job, Mike.
Thank you all so much. Thanks for watching.